Welcome to the second unit, section 1 of the molecular biology paper. The section 1 of the second unit is bacterial chromosome and plasmid. In this unit, we are going to see about how the prokaryotic genomes are present. How the prokaryotic genome is present as well as the structure of a plasmid. So these are the two things we are going to see in this particular topic of prokaryotic genome and plasmid. So prokaryotic genome. So everybody knows it. Prokaryotes means it's a underdeveloped nucleus. The prokaryotes contains underdeveloped nucleus. What do you mean by underdeveloped or pre-developed or not fully developed? It means that the genetic material is not covered or it is not placed in a membrane bound organelle. The genetic material is present freely. That is what the word prokaryotes means. So then prokaryotic genome means the genome is going to present in the cytoplasm itself. It is not going to be present inside the nucleus, what we are used to say in eukaryotes. So that is not going to be present in prokaryotes. So in prokaryotes, the genome is going to present in the cytoplasm itself. There are about more than 2000 prokaryotic genomes have been sequenced now. And then the structure of this prokaryotic genome is usually circular in nature. And in few cases, you can see some exceptions, like in Vibrio cholerae. So in that, it has more than one circular DNA. Similarly, Borrelia burgdorferi, which has one larger linear chromosome and 21 smaller chromosomes. So these are few exceptions, but more than 90% of the prokaryotic organism is going to have a circular chromosome, single circular chromosome considered to be as a prokaryotic genome. The total amount of DNA in prokaryotic genome ranges from 4,90,885 base pair in Nanochium equitans, an archaea that lives entirely within another archaea, to 91,05,828 base pair in Bradyrhizobium japonica. It's a soil bacteria. So the amount of base, the number of base pairs in a genome is going to vary from 4 lakh to 90 lakh. So 100 times there is a variation. It shows that that type of wide variety of prokaryotic organisms are present. That's what this variation in the base pair number indicates. Okay, so it is, even though it has contained different number of bases, all the bases are present in a circular chromosome form. The model organism of prokaryotic organisms is going to be usually a Escherichia coli, or simply we call it as a E. coli. And this E. coli usually contains around 4.6 million base pairs. Archaea and bacteria are similar in ranges of genome size. In some, already we saw that in some prokaryotic genome, the genome size is going to be very high. So this is about the size of the genome. Even the table, what you are seeing now, indicates that the size of the base pairs is going to be a very variable one. So it will start from like 0.49 million base pairs to it is going to present like 9.11 million base pairs. So this is how the number of base pairs varies. Similarly, the number of genes in the genome is also going to vary from one microbe to the other microbe. So if you see in Escherichia coli, you can able to see about 4,289 genes are present. Similarly, if you go to Nanochium equitans, the number of genes is going to be 536. So similarly, in the case of Mycoplasma genitalium, we have about 480 genes. And the genes is also going to be like more than 4,000 in the case of Escherichia coli. It is less than 500 in some species. So like its size varies, the number of genes is also varies between one bacteria to the other bacteria. So that is what we are seeing from this table. Each microbe has its own size of base pair as well as the number of genes. You can't correlate, you can't link. If the size increases, then the base has to be the gene has to be increased. In some cases, even though the size might be bigger, but the number of genes is usually found to be very less in number. So this is how the genome size as well as the number of genes varies from one microbe to the other microbe. And now you will see the structure of an E. coli, in which how the genetic material is going to present in the genome of the microbe, how it is going to present in a prokaryotic organisms. So we already saw that the prokaryotic genome is going to be circular in shape. Okay, so the circular genome, you can see this, it is going to attach with the RNA protein core. So in center you will see a RNA protein core, and this electron microscope is also shows that the RNA protein core which is attached with the genetic material which is attached to this particular RNA protein core. Then after the DNA molecule which is attached with this RNA protein core, these DNA molecules are further supercoiled. You can see here the DNA molecule is going to be supercoiled. Usually the diagram shows that it's a single strand, but actually the DNA is not the single strand, it's a double strand. So the supercoiling means what? Coiling of DNA upon itself. That is what it is called as a supercoiling. So here the DNA is going to be supercoiled here. So the DNA is first attached with the RNA protein core, and then it gets supercoiled, and then there is a doubt arises. There are two things we have to confirm here. One, the DNA is attached with the RNA protein core. The other one is the thing is that it gets supercoiled or not. So this experiment is actually carried out by a scientist known as Petitjean. So Petitjean carried out an experiment with the help of two enzymes, namely RNAs and DNAs. So he took this particular prokaryotic genome and treated with these two enzymes separately. That too partial hydrolysis. It is not allowed the enzyme to act for a longer period of time. He makes the enzyme to act on this particular genome for a short period of time. So it will cause only partial hydrolysis. Partial hydrolysis means only few of, say if you add RNAs, it will degrade only few amount of the RNA. If you add DNAs means it breaks down only few amount of DNA molecule. So when he treat this coiled folded genome to RNAs, he found that at certain regions the supercoiled size of the DNA, loop of the supercoiled DNA size is increased. When he treat the core with DNAs, you can see that the supercoiled nature of the DNA is removed. So this indicates that when you make a nick in the DNA, because the supercoiling is nothing but making twist upon DNA. So when you make a single strand or double strand nick in the DNA, the supercoiled is removed. So when you make a cut by DNAs, the supercoiled is removed. So that is why you can see the double helical structure of the DNA when you treat the genome with DNAs. Similarly, the DNA is attached with protein RNA core. So when you degrade RNA, it affects the interaction of DNA with RNA protein core. So the interaction point is released, so the loop size is increased. So that is how the Petitjean proved that the prokaryotic genomes are present not in a relaxed manner. The DNA alone is present. The DNA usually folded by itself in the supercoiled structure, and then it is also attached with the RNA protein core. So this is how the prokaryotic genome is arranged or present in the bacteria, in the prokaryotes. That is what the proposal carried out by this Petitjean, and this diagram is clearly indicates that. So this is the electron micrograph diagram where you can see the supercoiled DNA molecules which are attached with the center RNA protein core. So this is how the genome of prokaryote as well as the bacteria is present. Next, how the genes which are present in prokaryotes. Is there any speciality of the genes of prokaryotes when compared to eukaryotes? Is there any difference? Is there? I hope everybody knows that yes, there is a difference between gene organization in prokaryotes when compared to eukaryotes. Usually the genes in prokaryotes are usually called as a polycistronic in nature. So poly means everybody knows it's a many. Cistron means a gene. So polycistronic in nature is a multi-gene nature. So in prokaryotes, the genes are not present as a single gene. Usually the genes are present in clusters. So that is what leads to the new concept of operons. So in prokaryotes, the genes are present in a cluster in an operon format. So that is why it is called as a polycystonic in nature. If you take E. coli, there are about more than 3000 genes are present in the E. coli. Similarly, the repetitive nature is also going to be reduced in prokaryotes. The genes are usually unique in prokaryotes, not like in eukaryotes. In eukaryotes, genes are usually a single gene, and they are going to be containing intron and exon, so they are called as a split genes. But here in prokaryotes, the genes are present in cluster, they are unique in nature, they are polycystonic in nature. So it leads to the concept of operon in prokaryotes. So you can see here the example. See, if you take a tripoperon, you can see that there are about 5 genes are present in a cluster. In the case of E. coli genome, in the structure of a tripoperon gene, you have about 5 genes are present. When it gets transcribed and you get an enzyme from that, you will get 5 different polypeptide chains. So this is how the genes are present in a clustered manner in prokaryotes. But when you go to eukaryotes, okay, for example, in the case of yeast, you can see that the genes are present in different places, in different regions, different chromosomes, 4th chromosome, 5th chromosome, 7th chromosome, and 9th chromosome. And these again transcribed individually, and you will get 5 different polypeptide chains from 5 different locations, 5 different regions. So that is how the eukaryotic genes are usually called as a single gene concept, whereas in prokaryotes it is going to be a multi-gene concept. So the mRNA of prokaryotic organisms are usually called as polycystonic in nature, whereas in eukaryote it is called as monocystonic in nature. So whenever we call it as a operon, what do you mean by operon? Because the operon, the expression of the gene is mainly controlled by the operator. So that's how it gets the name of operon. So operating. It is not like a constitution gene that is always expressed. Its expression is controlled. Okay, so usually these genes are containing a specific structure, which contains usually promoter, operator, and structural genes. So this promoter, operator, and regulator genes are usually called as a regulatory genes, whereas these genes which are going to provide enzymes or other molecules are known as structural genes. So each operon will contain a regulatory gene and a promoter region, an operator region, and structural genes. Okay, so this is what the general structure of an operon. So this is how usually the genes are present in prokaryotic organisms. So whenever you take a prokaryotic organism, you can see the genes are present in a clustered manner. So with this cluster of genes, before we have operator and promoter, and these operator promoters are under control of regulatory genes, and these regulatory genes have their own promoters and regulators will also present. So as a whole, we call them as a operon. Okay, then next question arises to us. What is that question? Why these genes in prokaryotes are present in a clustered manner? Is there any advantage? Is there any advantage for the presence of this operon genes in eukaryotes? Anybody knows that? Yes, I hope everybody knows it. That is, the genes are clustered because to reduce the size required for the number of genes present in a smaller area. So if you see the same diagram, if I will draw here genes, if they present in different, that is in different chromosomes, what will cause the difference? That what I told is, if genes present individually, it will take large amount of space. How you can prove that? You can prove that here. You can see that if you take a gene, okay, so if this is the size of the chromosome, okay, so if you have a gene here, you can see that structural genes. Okay, so now you will see that the genes are, say, A gene is present, B gene is present, and C gene is present, and D gene is present. Okay, the same size. If you have individual gene has to present, so this is the regulatory region is required. Okay. So if you need a new gene has to be present individually. Then what will be the sizes required? For A you require this much length, and for B also you require this much length. Okay, then you require for C, and then for D. So you require the same type of length of the gene. So if you combine all these in a single line, okay, then think the length, what is the required length of the gene. See, so if you want to place 4 gene, you require the length. See, within this length all the genes are present, 4 genes are present, but now you can see there are about 4 or 5 times the length of the genome is increased if you want to present the gene individually. So this is how we are saying that the eukaryotic genomes contains in a multiple gene nature. So that is what the advantage of prokaryotic genes in a cluster form. So this is known as the prokaryotic gene nature. So next what we are going to see is plasmids. So what do you mean by a plasmid? Plasmid is usually called as a triple C molecules. What is triple C? Covalently coiled circular DNA. Okay, once again repeating it. Plasmids are simply called as a triple C. Triple C means what? Covalently coiled circular DNA. So plasmid is also going to be a circular in nature. Okay, so this is also usually present as a piece of a DNA, and then this is present outside the genome. Okay, so this is not integrated into the genome. It is not called as a plasmid. So the circular DNA, when it is present apart from the genome, so it's sometimes called as the episome. So the plasmid, if a bacteria obtain any character from this plasmid, it is called as a episomal character. That is, the episomes are nothing but the DNA which are present other than the genome. That's why it is called as a plasmid. So generally plasmid means what? It is present outside the genome. It is not essential for the survival of a bacteria. A bacteria can survive without the presence of these plasmids. But why then plasmids are present in a bacteria? The presence of plasmids in a bacteria provides additional characteristics to the organism. So that is what the nature of plasmid. So why the plasmid is present means this is what the reason. So the presence of plasmid provides some additional characteristics, additional features, additional nature, additional capacities to the bacteria. So that is what the advantage of these plasmids. But the bacteria can survive without the presence of these plasmids. There are about more than 270 plasmids are identified in the E. coli alone. So think about the number of plasmids which are present even in other bacteria and other things. So if you see a plasmid, this diagram clearly indicates that. So this color, this indicates that the genome of a bacteria. But here the small circles indicates that plasmid. So they are present apart from the genome. So that is what the plasmid means. So the plasmids are present outside the genome. But each plasmid have several important regions. So if the plasmid has to survive in the bacteria means it has to have an initiation site for replication. And then it has to be has the controller regions, regulatory regions, and structural genes which are going to synthesize important proteins which are useful for the bacteria. So that is what the advantage of plasmid. So plasmid will have important gene structure, structural genes it has. It has an origin of replication site. It's like that it is having important regions. Then only it is able to survive in the bacteria. So that is what the basic structure of a plasmid what we are seeing here. So if you take any plasmid, if it has to survive in a bacteria, it needs this origin of replication. Because this origin of replication alone makes this plasmid to survive in a bacteria, when the bacteria undergoes division, this plasmid will also divide with the help of this origin of replication. So any bacterial plasmid will contain this origin of replication, and they will also have some structural genes, and then they also have some regulatory regions, controlling regions for this replications or transcription and translation. So they containing certain regions for the regulation of the expression of these plasmids. Okay, so this is what the basic structure of a plasmid. So when you draw a structure of a plasmid, you draw a circular DNA and then try to put origin of application and then regulatory regions and structural genes. You should not forget to write these type of things. Okay, so next we will see about the important nature of this plasmid types. There are about different types of plasmids we have. One is a resistant type of plasmid, fertility plasmid, killer plasmid, degradative plasmid, and virulence plasmid. What do you mean by this resistant plasmid? The resistant is nothing but an antibiotic resistance. So due to the presence of certain plasmids, the bacteria will have, it's an antibiotic resistance. We are very much familiar with bacteria with ampicillin resistant genes, tetracycline resistant genes, like that we have, no? So that resistance nature is provided because of the presence of certain plasmid in that particular bacteria. If you remove that plasmid from the bacteria, the resistance nature of the bacteria goes away. So the antibiotic resistance is only provided by certain plasmids, which are usually called as a resistance plasmid. Then another type of plasmid is known as the fertility plasmid. So this is what we already saw in gene transfer reactions. Okay, so this F plasmid. So when the plasmid is present in an E. coli, the E. coli is actively participated in the conjugation process. So if a bacteria has to participate in a conjugation process, this F plasmid is going to be a vital factor to play a vital role in conjugation. What is the advantage of conjugation? Because the conjugation leads to the transfer of genetic material from one bacteria to the other bacteria, or one bacteria from one generation to other generation. New characters can be transferred from one bacteria to other bacteria. So this is what the advantage of this particular fertility plasmid. So this is what this fertility plasmid is going to do. The next one is going to be a killer plasmid. So the killer plasmid, what they will do is, the killer plasmid, if they present in a bacteria, it can kill the other bacteria. This presence of the other bacteria can be lysed by the presence of this killer plasmids. The example for killer plasmid is nothing but a col plasmid, colchicine. The col represents, it is going to synthesize a protein which is known as the compound. Sorry, it's a compound which is known as the colicin. So this colchicine compound, the colicin compound, is going to block the multiplication process, mitosis. So it is an inhibitor. So it inhibits the mitosis so that the cell is unable to divide. If the cell is unable to divide, it is unable to survive. So the growth of the organism is affected. So that is how this killer plasmid will kills the other organisms. Then degradative plasmid. The bacteria will have the degradation nature. Some bacteria, everybody knows that the oil spills. It is called as superbugs. Okay. So like that, what is the speciality of these superbugs? The superbugs has an ability to lyse the oil contaminations in the seeds. So this nature of bacteria is arised due to presence of certain plasmid. So one of such plasmid is the tall plasmid. So the name clearly indicates that the tall plasmid is going to help the bacteria to degrade toluene. So due to the presence of this particular plasmid, the bacteria is able to lyse the toluene molecule. So it is so that it can utilize the toluene and survive in a medium which contains the toluene. So for us, it is going to be like a detoxifying bacteria because it detoxifies the toluene molecule. Then the other type of plasmid is going to be called as a virulence plasmid. So the virulence, as the name clearly indicates, that the virulence is the pathogenicity. So it is the ability, the disease-causing ability is provided by the presence of these type of plasmids. So the example for this plasmid is a TI plasmid, tumor-inducing. So here, those bacteria which contains this TI plasmid has the ability to induce cancer. For example, Agrobacterium tumefaciens is able to induce ground gall diseases in the dicotyledon plants. So the presence of this Ta makes this bacteria as a tumor causing bacteria. So the induction of a tumor itself is caused by the presence of a simple plasmid. That is what the Ta plasmid means. Okay, so this plasmid will have different nature. As I already told you, this is usually present out of chromosomes, so that it is sometimes called as a episome. The plasmids are called as episome nature, and they can also merge with the genome in some conditions, and that condition is leads to the formation of new type of bacteria in the future. It can also maintain its copy number, non-integrative plasmids. When it is integrated into the genome, it is called episome. When it is not integrated, it is simply called as a plasmid. So, due to this type of presence of plasmids, the organism is going to have some special characteristic. As I told, it may have some resistance nature. Here you can see in this example, due to the presence of certain plasmid, the organism is going to become a tetracycline resistance organism. So this is what I already told you, that plasmid will have certain extra character. It provides some extra character to the organisms. So here you can see that the properties of plasmids. The plasmid have its own property. For example, copy number. So the copy number is that in any bacteria of a particular species, of a particular strain, it will contain the number of plasmid is uniform. Okay, so they are maintaining that number of copies. Say for example, if in a E. coli, say a particular plasmid, PBR, some plasmid is natural plasmid, so the 2 millimicron circle or any other plasmid, natural plasmid is present in a bacteria, it maintains its number. That is known as the copy number. So the number of molecules of an individual plasmid that are normally found in a single bacterial cell. Then the conjugative nature. No, I already told you, if F plasmid is present, then the bacteria has this conjugative nature. So that type of nature is provided by the plasmids. Similarly, in some cases, the nature of a plasmid helpful for the survival of an organism. I already told you, in a minimal medium. So if a medium contains high amount of chemicals like toluene and other toxic substances, if any cells has to survive, it has to have such plasmids so that it can withstand the particular chemical. So it needs that particular plasmid. So plasmid helpful for the microbe to survive in a particular environment. So even the compatibility, when you want to grow, or say in the genetic gene technology, or say in the recombinant DNA technology, if you want to clone a particular gene in a particular plasmid, you should know that whether the plasmid will survive in a particular bacteria. Not all the plasmids are survived in a particular bacteria. If any plasmid has to survive in a particular bacteria, it has to have some compatible nature. I hope you know that. I already told you that. For example, the origin of replication is one example. If any bacteria or any plasmid want to survive in E. coli, then at minimum it should have that origin of replication which is recognized by the E. coli DNA polymerase. Then only the plasmid can able to survive. So like that, the compatibility of plasmid is also a vital role for the property of a plasmid. The next one is best characterized. Even some plasmids are known as the shuttle plasmids or shuttle vectors. Okay, why it is called as shuttle? Because it can survive more than one type of bacteria. And as I already told you that how it is possible for a plasmid to survive in two different host organisms might be that it has origin of replication which can survive, which can present in both the organisms. So this, for example, if a vector has to survive in E. coli as well as in Saccharomyces cerevisiae, that means that the plasmid will contain origin of replication of E. coli as well as origin of replication of yeast. So since it has both the origin of replications, it can survive in both the organisms. So that is how the vectors is called as a shuttle vectors, because it can survive in two different hosts. That's why it is called as a shuttle. Similarly, we have about there are so many different types of plasmids, and then the plasmid will provide an immense help for the industrial production of many biomolecules. Many proteins are synthesized only with the help of this particular plasmids, and several gene functions are studied with the help of this plasmid. So we have an immense applications of biological important molecules with the help of this plasmids. Okay, so this is about the end session of this particular section 1 of the second unit. If you have any questions, you can contact me through the mail ID sivabio@gmail.com, or you can contact me over phone at 9865866168. Okay, so if you contact me and you will post queries to the mail, I will try to solve your questions. So these are the books we are utilizing it for the preparation of this presentation. So you can also refer these books to understand about this particular bacterial chromosomes and plasmids. So with this, I conclude the first section of second unit of molecular biology paper. Thank you. Thank you.